11th class will be protein synthesis, and this is divided into two parts. 11a is going to be the genetic code in tRNAs. 11b will be mechanism of protein synthesis, initiation, elongation, and termination, and then antibiotic inhibitors of protein synthesis, and coupling of transcription and translation. So this is the third time we've seen the central dogma of molecular biology, the flow of biological information, DNA to RNA to protein, and we've talked about replication, transcription, and now we'll do translation, where we discuss how this triplet code of nucleotides gets converted into an amino acid sequence. The genetic code in tRNAs, we have a triplet genetic code, and we have 20 amino acids that we need to code. So if we used, rather than a triplet, if we used a dimeric genetic code where we use two nucleotides to code for amino acids, 4 squared is 16. We get 16 permutations of two nucleotides, and that won't cover our 20 amino acids. So we need a triplet nucleotide code in order to cover the 20 amino acids. 4 to the third gives us 64. So here's our 64 combinations of the triplet genetic code. One of the combinations, AUG, is a start codon, and that codes for methionine. We have 3 codons that are stop codons. So UAA, UAG, and UGA are stop codons. And then the other 61 triplets are used to code for amino acids. Some interesting things to notice here. The way you read this is the first position. The U is the second position, so everything in this box is U and U. So UUU is phenylalanine, UUC is phenylalanine, UUA is leucine, UUG is leucine. So this is called degeneracy, where we can have more than one codon that codes for a particular amino acid. Notice UUU and UUA code for different amino acids, but both of these amino acids are hydrophobic. So if we change what's in the third nucleotide of a codon, we will still get a hydrophobic or a hydrophilic amino acid. So this is a case with leucine. All 4 of these codons code for leucine. So the differences in the third nucleotide of the codon, they're all CU, 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 leucine. Here's isoleucine, AU, 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 AUU, AUC, AUA are all isoleucine, and then the AUG is the methionine. So this is not unusual that within the degeneracy, the only difference is the nucleotide that's in the third position. Same with serine, same with proline, same with threonine, same with alanine, same with arginine, same with glycine. So that's sort of a pattern here. That also means you can have a mutation of the DNA sequence that amounts to a mutation in the third position of the codon, and you don't change the amino acid that's being coded. So that would be a silent mutation. In this case, you could get a mutation from a U to an A, but you still code for a hydrophobic amino acid, so maybe that's not so bad. So very interesting code. Degeneracy of the genetic code. So methionine only has one codon. If this is at the start of the coding sequence of message, this is the start codon. If it's in the middle of the coding sequence, it's just for a methionine. So methionine, it's both a start codon and a codon for all methionines, regardless of where they are in the reading frame. Tryptophan has one. So only 2 amino acids have only one codon. Asparagine, aspartate, cysteine, glutamine, glutamate, histidine, lysine, phenylalanine, and tyrosine have 2 codons. Isoleucine has 3. Alanine, glycine, proline, threonine, valine have 4. And arginine, leucine, serine have 6 codons. Mitochondria have a few variations of the usual genetic code. Back here I said the almost universal genetic code, and I said almost because of the genetic code of mitochondria is slightly different. Known codon variants in mitochondria. So the normal code assignment is UGA is a stop. In animals, in vertebrates, UGA codes for tryptophan. In Drosophila, which is not a vertebrate, UGA codes for tryptophan. Yeast, tryptophan, tryptophan, tryptophan. A plus means that it codes for the normal code assignments. So these, when I say Saccharomyces or the vertebrates, these are the mitochondria in these particular animals. These are the mitochondria in the yeast. So the yeast and the animals, their nuclear genetic code is the normal codon. It's the mitochondria within those organisms that have a slightly different genetic code. UAA, it's normally isoleucine in animal mitochondria. It codes for methionine, and Drosophila, it codes for methionine. Saccharomyces and Torolopsis, it codes for methionine. The plus means it's the normal code assignment. AGA, AGG, normally it's arginine. In vertebrates, it's a stop. So we lost a stop here to tryptophan, but we gain a stop here. And then Drosophila, this codes for serine. All of these organisms, their mitochondria, have the normal code assignments. So you can see how this works. So those are minor changes within the genetic code in mitochondria, and that's why I didn't call it the universal genetic code, but it's very, very close. Here's our transfer RNA that we saw earlier. These are some of the invariant nucleotides that you have in each one of these arms. So you have an A, purine, modified G, a G, and an A, and there's also dihydrouracil in these arms. It's the D arms. Contains 2 or 3 D residues at different positions. Here's our amino acid acceptor arm on the 3' end, CCA, and there's a purine here. Anticodon, we have a pyrimidine, a uridine, another pyrimidine, and then we have our 3 nucleotides for the anticodon, and we're going to see how the anticodon interacts with the messenger RNA here in a minute for translation. Here's our extravariable arm, variable not present in all tRNAs. Here's our T-Cy-C arm. A U here is invariant. This is a 2-dimensional structure of the tRNA, and we call this a clover leaf. The 3-dimensional structure actually looks more like an L, so this is the space-filling 3-dimensional structure. This would be the CCA, amino acid acceptor. Here's our anticodon, and here we see our base pairing, short links of base pairing within here and there and there and there. So that's a double helical type structure. You can see the helical type structure here, helical type structure there, helical type structure here. The tRNA has a domain for the recognition of the codon and for attachment of the correct amino acid. So the correct amino acid is going to the CCA acceptor. Here's our anticodon loop. Here's our codon. This is the triplet that's on the mRNA. This is 5' to 3'. Remember, all base pairing is anti-parallel. So if this is 5' to 3', our base pairing has to be 5' to 3' in the other direction. So this is the 3' direction. This is 5'. This is 5'. This is 3'. Here's our base pairing interactions. Now we're going to have a phenomenon called wobble. And what wobble does is the wobble base of the anticodon determines the number of codons a tRNA can recognize. So even though we have 61 different coding triplets in the genetic code, we don't have 61 tRNAs. We have something more like 30 some odd. In different organisms, there's different tRNAs, but we have to have at least 31 tRNAs to recognize our 61 triplet codes. So one codon is recognized. If here's our codon, X is any nucleotide, Y is any nucleotide. The third nucleotide is G. The anticodon has to be a C. So G and C in the third position. This is the 3' position. This is the 5' position. Only one tRNA will recognize this codon. The same if this is a U and this is an A. So one tRNA recognizes this triplet. One tRNA recognizes this triplet. 2 codon recognize it. In the tRNA, this is a U. It can recognize an A or a G. If this is a G in the anticodon, this can recognize a C or a U. Remember that in our codons with a particular amino acid, its codon mostly varied in the nucleotide in the 3' position. For instance, we'd get a UUC, UUG, and those would code for the same amino acid. So that means one tRNA can recognize both of those codons. 3 codons recognized. If the anticodon in the 5' position has an inosine, it can recognize 3 different nucleotides in the 3' position of the codon. So here is the 3' position of the codon. Here is the 5' position of the anticodon, and this tRNA molecule is basically slightly flexible, and that allows different base pairing between what's in the 1 position of the anticodon and the 3 position of the codon. So by wobble, it means that this is actually slightly flexible and allows us to have alternate base pairing to the classical Watson-Crick base pairing. Very interesting. Inosine in the 5' anticodon position of the tRNA allows one tRNA to recognize 3 codons. Now the 3 codons it recognizes are codons for the same amino acid. This does not allow a tRNA to recognize a codon for another amino acid other than the amino acid that it accepts in the CCA position. Because of wobble, an organism needs fewer than 61 distinct tRNAs. Theoretically, 31 tRNAs is the minimum needed to read the 61 codons. So here's an I and an A, an I and a U, an I and a C. It's amino acyl tRNA synthetases. These are the enzymes that add the amino acid to the 3' end. So here's our cloverleaf-shaped 2-dimensional tRNA. We have a CCA. Here's the phosphodiester bond between the C and the A. Here's the 3', what was the 3' OH, and amino acyl tRNases make ester bonds between the alpha carboxyl group of the amino acid. This was the alpha carboxyl group of the amino acid, and the 3' OH group of the ribose. And so here is our amino acyl group. So this is the amino acid attached to the 3' A. In the case of alanine, if this is alanine, all this particular tRNA needed for recognition was a G in this position and a U in this position, and it would put an alanine on the 3'. If we have other base pairing in here, we will put other amino acids on the 3'. Linking that shouldn't be that linking an amino acid to the tRNA. There are 20 amino acyl tRNA synthetases, which means these tRNA synthetases have to recognize several different transfer RNAs. So there's an amino acyl tRNA synthetase for each amino acid. So we had 20 amino acids, 20 tRNA synthetases, and some of these are recognizing multiple tRNAs. So here's our amino acid. Here's ATP. Amino acyl tRNA synthetase makes the bond between AMP and the amino acid. So this is an activated intermediate. So here's our amino acyl AMP. This is the activated intermediate. It reacts with the tRNA, and this tRNA is represented where here is the C, and there's the A, and here's the 3' OH group, and the 3' OH group, which is right here, attacks this carbonyl and gives us an ester, gives us the ester bond, and AMP leaves. Factors that determine the specificity of tRNA synthetases. Specific anticodon in the tRNA, that's true in some tRNAs, it's not true in others. Other recognition regions, the base next to the CCA in the acceptor stem. Single base pair in acceptor stem, that was the G and the C that I showed you earlier. 3D structure of the tRNA synthetases. Protein synthesis has 3 steps: initiation, elongation, and termination. So initiation in prokaryotes, here's our 50S subunit, the large ribosomal subunit, the small ribosomal subunit. This together makes a 70S ribosome dimer. Here's our inactive 70S ribosome. Initiation factor 3 binds to the small subunit. 50S dissociates. Now we have GTP and initiation factor 2 and F-methionine tRNA in prokaryotes. This is our initiator tRNA, and methionine is an amide. So the amino group of methionine and formate have formed an amide. So that's why it's called F-met, formylmethionine tRNA, and this is only used for initiation. Plus initiation factor 1. So here's the F-met tRNA. Here's initiation factor 2 plus initiation factor 1. And now we've bound the anticodon of this particular tRNA to the AUG of the messenger RNA, and that's our start signal. Upon making this complex with the small ribosomal subunit and the tRNA, the 50S subunit binds, and our initiation factors and GDP and inorganic phosphate leave. So the process of making our initiation complex hydrolyzes GTP. The tRNA resides in what's called the P site, which means peptidyl site. This is the A site, which is going to accept another amino acyl tRNA, and it will bind to its triplet code, and then we'll start peptide bond formation. So that's initiation in prokaryotes. Initiation in eukaryotes, this is our small ribosomal subunit, and it interacts with the initiator tRNA, which has a methionine on it. At this point, the mRNA binds, so the mRNA binds, and this, what we see as the bulb at the end, that is the cap, and the cap binding protein. So the ribosomal subunit recognizes the cap. We still have the methionine initiator tRNA in this particular structure. And then the ribosome and the initiator tRNA, they move down the mRNA until they find the first AUG. So initiator tRNA moves along the RNA searching for the first AUG. Initiation factors dissociate and the large ribosomal subunit binds. So here we have an intact ribosome. The methionine is in the P site. Notice this is EPA. The methionine is in the P site. Amino acyl tRNA, this is the next amino acid that's going to be in the polypeptide chain after methionine. It binds to the A site. So this is, we could call that an acceptor site or the amino acid site. So the amino acyl tRNA binds to this site. We transfer with an enzyme that's called peptidyl transferase. We transfer the methionine to this amino acid and we make a peptide bond. And now the methionine and the amino acid, they're covalently bound to the second tRNA. The initiator tRNA no longer has an amino acid on it. Notice that the ribosome has started to move. And we'll talk about these next steps in the etc. We'll talk about those in L11b, where we talk about the mechanism of the protein synthesis. So this is the first peptide bond formation. Here's our dipeptide. It's hooked to the second tRNA, and the initiator tRNA is now empty. This is our assembled functional ribosome that has the mRNA. So here's our mRNA. Here's the 5' end. Here's the 3' end. This is the amino acyl tRNA that's in the A site. This is the amino tRNA that's in the P site. This is the tRNA that's in the E site. This one is going to exit. It'll exit from this site, and then the ribosome will move down one triplet codon. This tRNA will now be in the E site. This tRNA will be in the P site, and we'll have a new amino acyl tRNA that binds to the A site. So again, we will see this mechanism during L11b. So this concludes L11a.